Today we are going to talk about another topic in the immunology cellular components. We are discussing today about the differentiation of the T cells. So the differentiation of the T cells occurs in various body system. It starts from the bone marrow, then thymus, then lymph node, and it ultimately shows its effects up to the peripheral bloods. So now we'll discuss about the series of steps that occurs in the differentiation of the T cells, and we'll also talk about the positive selection, negative selection, the role of the cytotoxic T cell, uh, the macrophage lymphocyte interaction, the regulatory T cell, and IPEX syndrome. The IPEX syndrome stands for immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome, IPEX syndrome. So first of all, you have to know the indicators. This red indicator I have shown here is of CD8, and the blue indicator is of CD4, and this black indicator is of T cell receptor which binds to MHC1 or MHC2 molecule. So first of all, you have to know that we have the T cell precursor in the bone marrow. So this T cell precursor in the bone marrow eventually be converted into CD4 plus and CD8 T cells precursor in the thymus. So in the thymus or in the cortex. So in the cortex of the thymus, the T cell precursor which are present in the bone marrow will be converted into CD4 plus and CD8 plus T cell precursors. So there occurs the positive selection. There occurs the positive selection. The positive selection occurs in the thymic cortex, and I'm going to explain a bit more about the positive selection. So the T cell expressing the T cell receptors capable of binding self MHC on the cortical epithelial cells survive. So this is the T cell. This is the T cell positive selection. So the T cell expressing T cell receptor capable of binding self MHC on the cortical epithelial surface survive. So that means those T cell which are not expressing T cell receptor and are not capable of binding the self MHC on the cortical epithelial surface will not survive. They will be killed by the natural killer cell. So we have the CD4 plus and CD8 plus T cell precursor in the cortex of the thymus from the T cell precursor in the bone marrow. So now from the CD4 plus and CD8 T cells precursor, it is differentiated into CD8 plus T cell and CD4 plus T cell in the medulla of the thymus. So you have to know this. So again I am going to explain. So we have T cell precursor in the bone marrow, and in the thymus, in the cortex of the thymus, we have CD4 plus CD8 plus T cell which will be converted into respectively into CD8 plus T cell and CD4 plus T cell in the medulla of the thymus. And we discussed about the positive selection. Now let us discuss about the negative selection. So negative selection occurs in the thymic medulla. So the T cells expressing the T cell receptor with high affinity for the self antigen undergoes apoptosis or become regulatory T cells. So tissue restricted self antigens are expressed in the thymus due to the action of the autoimmune regulators. So the deficiency leads to autoimmune polyendocrine syndrome. So the deficiency of this negative selection leads to autoimmune polyendocrine syndrome. So here in the autoimmune polyendocrine system, what happens? We see the features of the chronic mucocutaneous candidiasis, hypoparathyroidism, adrenal insufficiency, recurrent candida infection. So we see this in the autoimmune polyendocrine syndrome. So these all occur in the thymus, the medulla of the thymus. So eventually the CD8 plus T cell gets differentiated into cytotoxic T cell in the lymph node. So we have cytotoxic T cells in the lymph node. Let us discuss a little bit about the cytotoxic T cells. The cytotoxic T cells have various functions. The cytotoxic T cells kills virus infected, neoplastic, and other various functions. It does other functions like donor graft cells by inducing the apoptosis. So it kills virus infected cell, it kills neoplastic cell, and it can also kill the donor graft cell. So in the donor, when there is that any transplantation, we use the immunosuppressant. So it releases cytotoxic T cell releases the cytotoxic granules that contain profound protein like perforin and granzyme beads. The cytotoxic T cell have CD8, which binds to MHC1 on the virus infected cell and induces the apoptosis. So from CD8 T cells we have the cytotoxic T cell, and from the CD4 T cell we have the helper T cell. So the differentiation of CD4 T cells to helper T cells also occur in the lymph node. So helper T cells will be differentiated into T helper 1 cell, T helper 2 cell, T helper 7 cell, and T regulatory cells. So let us talk about the T helper 1 cell. The differentiation of the T helper 1 cell from the helper T cell is aggravated by the interferon gamma and interleukin 12, and it is inactivated by the interleukin 4 and interleukin 10. So talking about a T helper 1 cell, I want to talk more about macrophage lymphocyte interaction. So you have to know about macrophage lymphocyte interaction. So these various T helper cells, TH1, TH2, TH17, and T regulatory cells secrete the respective cytokines. I will discuss that in detail in this video later on. First, let's talk about the macrophage lymphocyte interaction. So T helper 1 cell secretes interferon gamma, which enhances the ability of monocyte and macrophage to kill microbe they ingest. So this function is also enhanced by the interaction of the T cell CD4T L with CD4T on the macrophages. So macrophage also activate lymphocytes via the antigen presentation. So this is the interaction between the macrophage and lymphocyte. So the helper T cells also differentiate into T helper 2 cells. So this is enhanced by the interleukin 2 and interleukin 4 and suppressed by the interferon gamma. So the differentiation of T helper 17 cell is enhanced by the transforming growth factor beta, interleukin 1 and interleukin 6, whereas it is inhibited by the interferon gamma and interleukin 4. Whereas the T regulatory cells, the transformation of helper T cells to T regulatory cells is enhanced by the transforming growth factor beta, interleukin 2 and inhibited by the interleukin 6. So this T helper cell, TH1, T helper cell 2, T helper cell 1, T helper cell 17 and T regulatory cells secrete this respective cytokine and they have their respective function. So first of all, I will talk about the T regulatory cells. So the T regulatory cells helps to maintain the specific immune tolerance by suppressing CD4 plus and CD8 T cell effector function identified by the expression of the CD3, CD4, CD25 and FOXP3. So T regulatory cells is activated regulatory T cells which produces the anti-inflammatory cytokines like interleukin 10 and transforming growth factor beta. So I want to talk more about IPEX. The IPEX syndrome, it is known as immune dysregulation, polyendocrinopathy, enteropathy and X-linked syndrome. What happens in this IPEX syndrome? There is genetic deficiency of FOXP3. So it will be the autoimmunity. So it is characterized by the enteropathy, endocrinopathy, nail dystrophy, dermatitis or other autoimmune dermatological condition which is associated with the diabetes in the male infant. So previously I have told that that T helper 1, T helper 2, T helper 17 and regulatory T cells secretes the respective cytokines and they have their respective function. So we'll discuss that in detail also. So T helper 1 cell will secrete interferon gamma and interleukin 12. So interferon gamma and interleukin 12 helps to activate macrophage and cytotoxic T cell. So it occurs in the peripheral blood. So T helper 2 cell will secrete the interleukin 4, interleukin 5, interleukin 6, interleukin 10 and interleukin 13, which will activate the eosinophil and increase the amount of immunoglobulin E. So T helper 17 cell will secrete interleukin 17, interleukin 21 and interleukin 22, which will induce the neutrophilic inflammation. The T regulatory cells, however, releases the transforming growth factor beta, interleukin 10 and interleukin 25, which is also known as the anti-inflammatory cytokines, which prevent the autoimmunity, which maintains the tolerance of our body. So today in this chapter we discuss about the differentiation of the T cell, we discuss about positive and negative selection, we discuss about the autoimmune polyendocrine syndrome, and also we discussed about the macrophage lymphocyte interaction, cytotoxic T cell, regulatory T cells, and also we discussed about the various cytokines secreted by the T helper cell, T helper 1, T helper 2, T helper 17, T regulatory cells, and also we discussed about their functions. I hope you understood this topic on the differentiation of the T cells. Thank you.